Thank you very much for inviting me to present a little bit of my work here. And today I'm going to tell you a little bit of the work I've done over the last few years studying the rapid evolution of plants that have become invasive in new environmental ranges. I want to focus on a particular special case of a plant invasion. But I think before we get into the details of the presentation, I mean, I don't need to remind you that one of the most dramatic changes in the global environment has been the redistribution of plants and animals all over the world. So for example, in Europe alone, there's 12,000 species of plants and animals that have become invasive. That means that they have moved beyond their native range and now they are found everywhere. And, you know, we've been spending a lot of time and effort trying to control the spread of these invasive organisms, and we miserably failed on this. So it's easy to see how, despite our best efforts, the world's biota has already changed in a really massive and probably an irreversible scale. Now, biological invasions is a phenomenon that has really global proportions. You can see in these figures the area covered by invasive species and the number of species in different biogeographic regions of the world at the continental scale, Africa, North America, and Asia. And you can see by the units that these are really large scales. And the movement of species between these geographical regions is very complex. You know, take Europe, it receives migrants, invasive plant and animal species from all over the world. And because in part of this massive scale, introduced species represent probably the second most important threat to local biodiversity nowadays. Maybe just second to the transformation of the habitat due to human activities. So, I mean, it's no surprise that because of these characteristics, biological invasions are often a source of concern. But I want to argue that beyond this severe cost of biological invasions, non-native species give us a really special opportunity to study fundamental questions in biology. So what we can do is take advantage of this unplanned situation or unplanned experiment to look at how different species of animals, plants, and microbes respond to the ecological challenges that are associated with arriving to a novel environment. And one of the nice things about these invasions is that you often have replication either in space or time of the same non-native species arriving multiply. So taking advantage of this, part of my research in the last few years has been trying to address this type of simple but fundamental questions in biology using biological invasions. I want to understand, for example, how can populations that have never experienced a particular set of biotic and abiotic factors adapt and not only persist, but also thrive there. I want to understand how these invasive populations manage to do such a trick. Do they do it by recruiting genetic variation that already exists in the population, or do they need to wait for mutation to introduce new mutations before they can respond? And a particularly perhaps a non-traditional question that I'm interested in is whether actually biological invasions could act as a perhaps minor but non-zero contributor to biological diversity. And there's, I don't know if you guys have seen this book by Chris Thomas from the University of Oxford, which put forward this highly controversial hypothesis that because of this global change, biological invasions, invasive species, might not be just a part of the ecosystem, but might play an increasingly important part of new ecosystems. So he argues nicely, but controversially, of whether these inheritors of the earth are these translocated species. And we have nice examples how in the past, after massive global changes, there's been a reorganization of the world's biota. Now, one of the consequences of this global translocation is the emergence of new opportunities for hybridization between populations that historically have been isolated. So we have cases in fish, in birds, and in plants where closely related species, but that occur in different geographic regions, come into secondary contact, and this creates an opportunity for new combinations to arise. And I think in plants is particularly relevant, this secondary contact, because if you look at closely related species of plants, one of the main mechanisms by which these species stay apart is through these prezygotic barriers. That means barriers that prevent the gametes to come into contact and form offspring. Okay, so things that happen before mating is mostly what isolates species in their native range. And this translocation, this geographic meeting of isolated species then breaks down an important barrier for that. So I want to now give you a little bit of more of detail on what kind of hybridization I'm talking about. So hybridization is basically exchange of genes between two groups of individuals that are genetically differentiated. So this can happen both within species, if you have genetically differentiated populations that meet again, or between species. So in another common feature of biological invasions, in particular plants, is that populations of the same species that come from different parts come together multiply. So you have these repeated opportunities for hybridization. And when genetically distinct populations come together and form hybrids, then some interesting things happen. One of them is that you get this phenomenon that is well known in agriculture, which is called hybrid vigor or heterosis, which is that the hybrids display increased fitness, increased performance or survivorship, or increased seed production, for example. And it is there's different hypotheses of how you can get hybrid vigor, but they have to do with the relationship between genetic variants that occur in these populations. Either you sort of maximize the combination of alleles, or you create or one of the parents dominates in certain traits and another part in others. So it can be a good thing to mix genes across differentiated populations, but it can also come at a cost. Because of course, when you hybridize genetically distinct populations, then you break down whatever genetic combination that has been tested by selection, and you may end up suffering of a reduction in the performance or survival of these populations because of that breakdown, that recombination between genetically distinct genes. But now one of the things that I'm interested in is that all these are theoretical possibilities, but there is surprisingly limited evidence of what is the effect of hybridization or gene flow between genetically distinct populations during biological invasions. So if you look at biological invasions, do you get mostly positive effect or a negative effect? Now, the second consequence of hybridization is what happens when you combine different species. And now we're talking about secondary contact of biological entities that are reproductively isolated. There are many examples in invasive plants where hybrids are sterile. Okay, they cannot produce seed, but they're still capable to persist because, for example, they have the ability to reproduce clonally. And this secondary contact between species can be important, even if there's been genetic differentiation on the order of 4 to 8 million years. Plants retain the capacity to hybridize. Okay. This is data that comes from a nice review by Don Levin, where, for example, it shows that on average, plants have to be separated by 4 to 5 million years before they evolve sterility when they get crossed, and 8 to 10 million years before they evolve just simply incompatibility of forming a hybrid. And we know that those species that can go through these filters can then go on to become successful evolutionary lineages. And there's a number of classic examples from the literature where invasive species of, say, Tragopolon or Spartina or Senecio have escaped that sterility bottleneck and form fertile, self-sustaining species. So in the remaining of this talk, I want to talk to you a little bit on these two main questions that have shaped my research on plant invasions. First, I want to present to you work that we've done to look at what are the fitness consequences of this mixing within species during biological invasions. Okay? And then I want to tell you a little bit about what we have learned from this studying invasive plants of whether hybridization between non-native species can contribute in some way to biological novelty. Ultimately, what I care about is what can we learn about like fundamental ecological and evolutionary process by using biological invasions as an example. And now let me introduce the character of this story, which are the monkeyflowers, the genus Mimulus. Nowadays, goes with the horrendous name Erythranthe, but I'll stick to the old nomenclature and I call it Mimulus for the rest of the talk. This genus Mimulus has about 120 species that occur all over the world. It has centers of diversity in North America, South America, the Himalayas, and Australia. There's a fair amount of variation in ecological traits. And for the last 60 years now, they have been the focus of a lot of ecological and evolutionary research, mostly in North America by Vickery, a researcher that was very active in the 50s and 60s and studied a large number of these species. Now useful for a talk about hybridization. And by the way, please interrupt me if you have questions. They can intercross. Okay, although they are separated by a certain amount of time, they can still hybridize. Yes?
Why did they call it, why monkeyflower?
Ah, that's a good question. So how is your Latin or people's Latins? So can anybody recognize the name of the genus? Means mime. Yeah, and so there is no connection from the original description and monkeyflowers. It was a name that was picked up later, and some people argue that some species look a bit like a monkey face, or a mime face, or a face, a personate corollas. But actually, you have to stretch your imagination a little bit. It's a cool name. I love calling them monkeyflowers. So they can hybridize. This is like classic study between hummingbird and bee-pollinated species that produce all these cool hybrids. They have a small genome, which makes them amenable to doing genomic analysis. And they also have variation in ploidy, which becomes important later in the talk. There's growing genomic resources for this genus. Last year, we got a grant from the Joint Genome Institute to do the pangenome of Mimulus, which means sequencing lots of species as diverse as we can. And we're like 1 year in the project and starting to accumulate genomic information from species that we know very little about. And maybe a little bit less known, but very important for me and for this study, is that a few species have been very successful at escaping from the native range and becoming established in the introduced range. One of them is Mimulus gutatus, the yellow monkeyflower, which is native to Western North America from Mexico to the Aleutian Islands. It has been introduced in both Europe and in New Zealand. You can find it scattered somewhere. I'd love to hear what you guys know about Swedish monkeyflowers. And the other species is Mimulus luteus, which is a close related taxon from the west side of the Andes region, mostly Chile, Peru, Argentina, but a little bit in Peru. And this has been introduced in the 19th century into Europe. So let me now use this study system to tell you a little bit about, first, how does hybridization within species affects their performance? Is it a positive or a negative effect on the evolution of introduced populations? So this is a bit of a closer look at this first species, Mimulus guttatus. Is incredibly diverse in the term of habitat that you can encounter. You can find it from dry coast to the headlands that are permanently exposed to moisture, to inland populations that are extremely seasonal, and so on. There is variation in the life history. Some populations are annuals, others are perennial. Some are exclusively sexual and reproduce through seed, and others can combine sex and clonality to do kind of the best of both worlds. They've been introduced in different parts of Europe and New Zealand. This is a map made by one of my former master's students, where we looked at the suitability of habitat of these plants in different regions. And we found that, for example, the British Isles appears to be a good place for them, not so much where we are. But one nice thing, so I started working in this system when I just arrived to the UK. And one nice thing about studying these invasions in the UK is that there's a very good historical record of when and where do they first arrive. So you can find, by looking through this historical literature, that Mimulus guttatus, pictured there on the left, was first imported as a botanical curiosity in the 19th century. And it quickly became a favorite of Victorian gardeners. And you could find in seed catalogs material from different species to plant in your garden. So not only guttatus, Mimulus guttatus, or the yellow monkeyflower arrived, but also other species coming together and start hybridizing, probably in purpose by people creating new floral varieties that were particularly colorful or striking. We know exactly the year where this is first recorded. It was 1812, and it was reported from this part of the UK, or near Cambridge. And we know that about 20 years after the first record, we start seeing the first naturalized populations pop out all throughout the UK. It's a plant that prefers wet habitats, the sides of rivers or marshes or roadside ditches. It is widespread, as you can see in this density of occurrence records since the 1950s. Okay, it's found all over the place, in some parts more than others. It's locally dominant. That means when you find monkeyflowers, you tend to find lots of them, but it's relatively non-aggressive. So only when it reaches very, very high densities, it displaces the native flowers. Other than that, it just seems to be able to coexist fine. Now, what is really cool is that you can not only find where and when did it arrive, but you can also find out who brought it to Europe. It turns out that the person responsible for first discovering monkeyflowers, it was the first species of the genus that was reported, was this Prussian naturalist working for the Russian Tsar in one of the first expeditions to circumnavigate the world. And this guy, Grigory von Langsdorff, was conducting an expedition for the Russian-American company that owned Alaska at the time. And he was a naturalist on the boat and would pick up plants and animals on each trip. And if you read his diary, you can find that in July 14 of 1805, he was crossing a path that connected Beavers Bay with Ugadaga, which is currently called Dutch Harbor in the Aleutian Islands. And right there, he finds the monkeyflower. He brings those specimens to Europe, and Linnaeus describes them and ascribes a taxonomic name. And why am I telling you all these? Is because we have very precise historical record that suggests where these things might come from. So what we wanted to do in our in this part of the research was to test this historic hypothesis and see if we could gather genetic evidence that indeed the monkeyflowers that you have nowadays can be traced back to this Russian explorer. So together with a network of collaborators, we conducted a global sampling effort to get plants from the UK, New Zealand, Western North America, both in the native range, and in the introduced range, the Faroe Islands, everywhere where we could find monkey flowers. And key to this, to the connection with the history, we're able to go to Alaska and retrace with the help of local botanical experts. You know that to trace their ancestry to this Russian period, the exact paths and the exact areas where Langsdorff and his crew might have been finding these first monkey flowers. And then we took this about 500 samples from about 130 populations and genotyped them by scanning their genome at a relatively low density. And this allowed us to get a lot of markers to genetically compare these populations. And I'm going to skim through the details of the genetic analysis, but I'm very happy to talk about them with you. Basically, we found that these are the UK populations. This is native North America. These are, the purple are the Alaskan populations. And these are New Zealand populations. We can find some here and some there. And these are the Faroe. The blue ones are the Faroes. So what I want you to take from this is that the most widespread invasive populations are genetically distinct to the other populations. Not very obvious that they are closer to these Alaskan plants than this one. And it's not very obvious, even if you start looking at different ways to represent the data. But we have conducted a series of analyses, including like building phylogenetic trees or family trees of these populations and doing more complex demographic models that allows to test specific hypotheses of relationships. And we're fairly confident that the UK plants, at least, are most closely related to Aleutian plants than anything else. We cannot have like an exact correspondence with these populations that Langsdorff sampled, but there seem to be at least most closely related to that. And other thing that we were able to gather from this data is that invasions in New Zealand and Eastern North America are nested within the UK, which suggests that the UK has served as a bridgehead, as a stepping stone in the invasion process. The plants go from Alaska to the UK, and then from the UK to New Zealand, or to Eastern North America, and so on. So it highlights how invasive populations can act as sources for this. And they finally they have a relatively high level of genetic diversity if you compare to other regions. Not as much as in the whole range, but a fair amount. They have not been like genetically depauperate. Okay, so now that we know a little bit about the history of this invasion, let's look at what happens. And we know that they have high genetic diversity, and they have probably come from different parts of the range. Now we can look at what are the effects of crossing different genetically differentiated populations on the performance and fitness of these plants. And the way we did this was taking an experimental approach. And this is work by my former PhD student, Pauline Pantoja. And what Pauline and I did was to take 3 populations, one from California, one from Alaska, and one from Scotland. Okay, we wanted to have an introduced population from Scotland, one that was close to the source, Alaska, and one that was far from it in California. They were different not only in their geography, but also in their life history. The Californian plants behave as annual, the Alaskan plants are perennial, and the Scottish plants are perennial. And what we did was to take each of these populations and cross it against Scottish mom. Okay, so why do we want to cross this? Because we wanted to generate variation in the characteristics of these plants so that we can then put them in the field and look at how they do, depending on their origin. We wanted to homogenize to an extent the genetic background, but still introduce variation coming from these 3 areas. So we created first generation hybrids, and then we pollinate those plants with themselves and created F2, an F2 generation is very variable within each of those crosses. Then we put these plants in the in the field, in an experimental garden, and followed them for a year to measure how they grew, how many seeds they produced, how many clones they produced. So I'm going to now show you the results of that experiment. And it was a massive effort by Pauline following a few hundred plants. So in colors are the paternal parent of the cross. So the blue is the Californian one, the green is in the middle is the Scottish one, and the red one is the Alaskan one. The first panel is the number of stolons, which has to do with how well they are reproducing clonally. This is when do they flower, and this is how many flowers they produce. And you can see that each of these cross types behave very differently. For example, the Alaskan plants are good at cloning, but produce few flowers. The California plant line is bad at cloning, as we expected, but relatively lots of flowers. And the Scottish by Scottish cross seems to be good at doing a little bit of both. Okay, so now the question is how does this add up to the total performance of these plants in the field? And one of the complicated things, if you want to estimate the fitness of these plants, is that you have very different currencies. You have number of seeds and clones, right? You can either spread out laterally with these ramets, and you can also produce seeds. So how do you weight? How many seeds are worth one clone? So the approach that we took for addressing this complication in calculating total fitness was to use plant demography. So we built a life cycle of these plants, and we're able to estimate parameters that describe how much a particular population will grow or shrink depending on the seeds and clones they produce. So the summary of that approach is a single number, which corresponds to evolutionary fitness of that plant, and is now comparable between plants of different life histories. And we estimated the variation in this relative fitness, and found that when you include different components of fitness, the Scottish by Scottish cross, the introduced by introduced cross, has the highest fitness of either of them. Okay, so what does this mean? Okay, we have evidence that suggests that if you cross within the invasive range, the plants do well. But if you cross with the invasive range with something else, things don't go as well. So this is evidence of outbreeding depression, this loss in fitness that arises presumably because of the invasive populations are locally adapted. You know, so this is one possibility. Why do the inter-population crosses reduce fitness? Because you're breaking that local adaptation. There are other possibilities. Could be things that don't have to do with the environment where the plants grow, are simply genetic differences that when you bring them together cause problems. So this is called environment-independent genetic incompatibilities. But maybe one of the points I wanted to make with this study is that it is very important to look beyond the early generation of these hybrids and try to capture the performance of these plants in the field. If we have looked at the F1 generations, they display this hybrid vigor, and they seem to do well regardless of the environment. And it's only when you allow recombination to happen and you put them out in the field that you start noticing the differences. Okay, so I will then just briefly present the second part of the story, which is how does hybridization between species contribute, or to what extent contributes to biological diversity. And I briefly told you that along with Mimulus guttatus, other species were introduced into Europe in the 1800s. A second wave of invasion following closely Mimulus guttatus was this group of species from South America in the species complex Mimulus luteus, which includes species that are interfertile but phenotypically differentiated. They have very different flower colors, for example. And this breakdown of geographic isolation then enable hybridization between different species. And this hybridization has resulted in a beautiful mosaic of floral diversity that you currently find in extant populations of monkeyflowers. These are examples taken in the field, including the two parents, Mimulus guttatus, Mimulus luteus, and everything in between. So using genetic estimates, we calculate that about 40% of all monkeyflower, non-native monkeyflower populations are hybrids. Okay, so it's a non-trivial amount. But this is kind of weird because these hybrids are sterile. Okay, they are vigorous, they can clone, but they are sterile. They produce pollen grains, but these pollen grains are empty, as is shown there by the aniline blue lack of stain. And why are they sterile? It's because they suffer from their parents having a different chromosome content. Just like mules are sterile because they are the result of a cross between a horse and a donkey, and they have different chromosome numbers. These botanical mules are sterile. Nevertheless, they are relatively widespread and have been found in the British Isles since at least the 1870s. Now, I would love to hear your hypothesis of how they're getting around, but I think it's facilitated by people. So if you have a sterile hybrid, it might be ecologically important, it might be everywhere, but evolutionarily, it's a dead end, right? There is no fate, no future for that line because it cannot leave any fertile offspring. However, there is a way out of sterility that is commonly taken by plants when they are found themselves in this evolutionary dead end. And this is that they can get out of sterility by doubling their genome. And this is a massive mutation, probably the most dramatic mutation that exists in plants. When in a single step, you double up the number of chromosomes. And because the doubling of chromosomes rebalances the proportion of the two parental genomes, then this is often associated with full fertility, okay? And this is not just a theoretical possibility. It has been recorded in Tragopogon, Spartina, and Senecio, all of which have at least one invasive parent, okay? So, as you can guess from the title of the presentation, we found exactly this process happening in the British Isles. We found that sterile triploids, okay, with three chromosome sets, have given rise to fertile hexaploids, 6X, through this polyploidization event or polyploid mutation. And this new taxon is now pollen and seed fertile. Okay. And here's a nice picture from my collaborator, John Bailey, of the chromosomes painted in a fluorescent microscope. Okay, so this plant, which I call Mimulus peregrinus, is one of the handful of examples of species that have evolved in the last 150 years, which makes it slightly younger than Darwin's Origin of Species. Now, where does this plant occur? We first found it in 2011 in the region of Leith Hills in the southern uplands of Scotland. And since then, we found another population or a set of populations in the Orkney Isles in the north of Scotland. Now, when you have, and this is a picture of the two plants from these two populations. If you have two populations of this plant separated by a couple hundred kilometers with nothing in between, it raises the possibility that they have independently originated. But it's still not proof, right? It could be that some rare migration event has caused them to move. So how different are they? If you look at the floral and leaf traits, and you combine all of them in a principal component analysis that gives you an idea of the overall differentiation, you can find that the Orkney population in red and the Left Hills population in blue are somehow genetically differentiated. Statistically, you can't tell them apart, although maybe for the untrained eye is not straightforward to do it. But yet, this is not proof of their genetic relationship. So to look at how they are related, we did a genomic analysis using a neat trick called sequence capture, where you create probes or baits of DNA based on the genetic sequence that you get from the relatives. And then you use that to subsample the genome of these plants. And the goal for this is to simplify the complexity of the genome and to analyze thousands rather than millions of markers. So we're able to get information for about 1,200 genic regions and sequence these at a relatively high depth. And what this data helped us to do is to show that these two geographic locations represent two independent origins of the same species, Peregrinus, here and here and here, evolving from local triploids. So this is a curious but by no means unusual case of the same species evolving twice over the last 150 years. Okay, but I remember when I presented these results, somebody in the audience asked, well, okay, they share the parentage, but are they interfertile? Okay, if they are not interfertile, you cannot cross them, they're not the same species, because one of the definitions of biological species is that they can reproduce with each other. It might be that you get the same thing, but it actually are two biological entities. So we were interested in addressing exactly this question. And what we found that if you do within Left Hills crosses or within Orkney crosses, this is the proportion of germination of the seeds. And if you do inter-population crosses, you get about the same or maybe slightly more. If you look at pollen viability, this is more variable. There are populations that produce more viable pollen than others, but the interpopulation crosses is not different, which suggests that indeed these are biologically the same thing. Okay, so I'm now approaching the end, but before I finish, I want to address just one last question, okay? That maybe you are thinking as well, which is like, okay, you're having these entities that evolve relatively rapidly, but so what? Like, do they last? Do they go extinct? What are the capacity to persist? What is the evolutionary fate of these newly generated polyploids? And if you want to address that question, then you need to look at fitness, but you need to look at the fitness of these plants, not in a growth chamber, but in the field. So what my former postdoc, Sophie Mers, and the team did was to set up these experimental plots in the southern uplands, maybe 20 meters from where we first found the plants. And then it's a region that has naturally the triploid, the sterile Robertsi, and the hexaploid, the fertile Peregrinus. And then we transplanted 800 clonal replicates of both of these hybrid taxa and the parental taxa. And then we looked at how do they survive the winter. So the results of this study is summarized here. Okay, this is Mimulus guttatus, and this is the survivorship of that parent. This is Mimulus luteus, and that's a very low, actually 0 in the second year survivorship of the other parent. Here is the triploid, and here is the polyploid, the new species. So it looks like both hybrids, regardless of whether they're doubled up genome or not, survive much better these winter conditions than either parent. Now, you might think that this difference, although it's not statistically significant, maybe this suggests that the triploid would do better. But you need to remember that this one does not reproduce via seed, but this does. So, and that advantage is not captured there. So we haven't yet done that demographic analysis that I showed you in the first part to balance these things out. But my guess is that seed production, which is high in these monkeyflowers, will probably tip the balance in favor of the hexaploid. Okay, So with that, I've shown you experimental results that suggest that hybridization within the same species, but between populations in different regions, results in this phenomenon called breeding depression or reduced fitness of the interpopulation crosses. I've given you an example of how hybridization and whole genome duplication can trigger not only phenotypic change, but rapid speciation as well. And I hope I've convinced you that not only new species can arise, but they can arise multiple times and lead to the same biological outcome. And of course, there's many open questions that remain and that I hope to address in the future. I'm interested in to what extent is this outbreeding depression depending on the environment where you grow the plants. If we put the plants out here, is that advantage broken because it's in a completely new environment? To what extent evolution that happens after invasion is more important than the capacity of those plants to deal with different environments in the native range? And ultimately, what happens in the long term to these hybrid lineages, particularly in the context that this global change is increasing the opportunities for hybridization, not only for monkeyflowers, but for hundreds or perhaps thousands of other taxa. And yeah, with that, I'd just like to thank Pauline, Sophie, Stuart, Matthew, and the other people in my lab, my collaborators, Josh, Richard, and Ariel, and my funding sources. Thank you.